Hello and welcome to this lesson on the immune system. So here I have this diagram, a whole bunch of different cells. This is depicting hematopoiesis, which basically it just means white blood cell differentiation. So we're going to go through and label all of this from start to finish this way. And then we'll delve deeper into, you can see that these are divided into like two halves. We'll delve deeper into the two halves of the immune system. So, first of all of our white blood cells are produced in the bone marrow. So I'm just going to write that right now. Okay, fantastic. Get that out of the way. Now, everything starts here with what is called a hematopoietic stem cell. So, use a bit of a smaller pen here. I will write that out. Hematopoietic stem cell. Okay. And then, as I said, there's two halves to our immune system here, and I will just go ahead and label them right quick. So down here on the bottom, we have our innate immune system. Oops. Innate. And up here, we have our adaptive immune system. Okay. And I'll start down here with the innate immune system. So from our hematopoietic stem cell, we can differentiate into a myeloid stem cell. So okay. And from there, just going from top to bottom, you have a monocyte. Okay, which can differentiate into a dendritic cell. Or macrophage. Okay, then here you see these three guys circled in blue, and I guess also includes this guy, but you'll see what I mean in a minute. So circled here, we have a neutrophil. Then an eosinophil. And a basophil. I hope you can read my chicken scratch. I apologize. And then here we have a mast cell, which are related to basophils. But anyways, these guys are called granulocytes because they have a bunch of these little granules sitting in them. So the main important ones are these three, but there's also this mast cell that does have granules in it that come into play, so just keep that in mind, but I will write out granulocytes. Okay, continuing with the labeling. So if you have a myeloid stem cell and you throw in some thrombopoietin, I will write that out. Goodness. Thrombopoietin, you get a megakaryocyte. Which will then break apart and form platelets. Alright, then back to our myeloid stem cell. If you throw in some erythropoietin, then you end up with erythrocytes, or red blood cells. I don't like how I wrote that. Better. Okay. I think that's it for labeling with the innate immune system. Now we have the adaptive immune system. So going back to our hematopoietic stem cell, it can also differentiate into a lymphoid stem cell. Which then can differentiate into a natural killer cell, or NK cell, as most people shorten it. And so, okay, this is now the end of the innate immune system. So you see you have these guys all circled together. I'm not sure why this is included in that. But anyways, natural killer cell, even though it comes from a lymphoid stem cell, is really, it's a part of your innate immune system. So, and these guys are circled in purple because they are what would be labeled as agranulocytes. Because they don't have granules, unlike these guys down here. There are not any granules in monocytes or natural killer cells. And again, I'm not really sure why I included the lymphoid stem cell in this, but the two important ones are here. Okay, moving on. Continuing with our diagram here, a lymphoid stem cell can differentiate into a T-cell. T-cells can then become helper T-cells. Okay, and cytotoxic T-cells. All right. So then down here, getting to the last bit, lymphoid stem cell can become a B cell, which can differentiate into a plasma cell. All right, and so within your adaptive immune system, there's two different categories, and I'm going to say your adaptive immune system. So there's your cytotoxic immunity, also called cell-mediated immunity. So, but I just use cytotoxic. Either one should be fine. And then there's what's called your humoral immunity. So cytotoxic immunity involves all your T cells, and humoral immunity involves your B cells and plasma cells. And we will get into that later. Yeah, okay. So let's move on. So the innate immune system, we'll start here. Also called nonspecific immunity. So the innate immune system it's always active. So it's always doing stuff, it's always on the alert, but it can't target specific invaders. And it also can't maintain immunologic memory. So what does this mean? Yeah, it's always doing stuff, so it's always active. But it's non-specific, so if it recognizes something as foreign, it'll attack it. Very simple. And it doesn't necessarily, it doesn't remember what it attacks, but it doesn't necessarily need to. That's not the point. We'll get into that later with the adaptive immune system, but anyways. So within the innate immune system, many of the non-specific defenses are also non-cellular. So I have made many drawings here of non-cellular defenses, and we'll just go through them. So here I drew a whole hand, I don't know why, to depict skin. I suppose it's just like the most recognizable body part. So skin acts as a physical barrier, and it also secretes antimicrobial compounds like defensins, and also sweat, which I drew out here, you know, sweat on your armpit, which it really actually does keep your skin clean. And also mucus and mucous membranes, they trap pathogens. So for example, you know, sometimes you get mucus buildup in your throat that is caught by your cilia and can either be swallowed or expelled. So any of you guys out there that have postnasal drip, like me, you are quite familiar with this. Next up, there's tears and saliva. Contain lysozymes, which is an antibacterial compound. Then also, I mean, your stomach has stomach acid that kills most pathogens. Colonization of the gut also prevents overgrowth of pathogenic bacteria through competition, actually. So good bacteria will beat out the bad bacteria. The idea behind this is that your gut is already fully, if not mostly fully, colonized by your good bacteria, your own bacteria, and therefore there's no room for the bad bacteria to even start to multiply. So that's the idea of that. There's also then the complement system, which made very simply just punches holes in the cell walls of bacteria, which makes them osmotically unstable. So here I've made some holes in the cell wall of this bacteria. Osmotically unstable, you know, cell walls don't really allow for any kind of osmosis, like liquid, I'm going to say, transport between the wall. So as soon as you punch holes in there, suddenly osmosis happens. And then over here we have interferons, little proteins. These guys are given off by virally infected cells, so we have our little viruses here, that can help prevent viral replication and dispersion to nearby cells. So, you know, they're here, they go and attach to these little receptors on cell membranes of these cells, like basically warning them that there's an infected cell nearby. Be on alert. There's already a cell here, a little virus here. So, yep. those are the basic non-cellular defenses of your innate immune system. Okay, now here I told you we'll get into this, so here's cellular defenses of the immune system, and we'll go through these one by one. So, you have monocytes. Monocytes are just phagocytic cells that can differentiate, as we said earlier in our diagram, to dendritic cells or macrophages. So, monocytes in the bloodstream become macrophages in tissues, and for example, in central nervous system, you have microglia. In your skin, you have Langerhans cells. In your bone, you have osteoclasts. Those are all macrophages. So macrophages, what color should I use? How about, orange? Macrophages, they ingest pathogens and present them on major histocompatibility complex molecules, which we shorten to MHC molecules, and I will also get into those in a second. So, let's see what to write. Just pathogens and okay, and macrophages, they also secrete cytokines. Cytokines are responsible for inflammation and also further recruitment of other cells of the innate immune system. So I will do secrete cytokines. Cytokines, I think the most important thing to remember about them is inflammation, so we'll do okay. So the major histocompatibility complexes. Phew. There's two of them. There's two, MHC1 and MHC2. So MHC1, and I should do this in a different color, maybe green. MHC1 is present in all nucleated cells, and it displays an endogenous antigen, non-self proteins from within the cell, to cytotoxic T-cells. So endogenous antigen meaning like a substance, usually a pathogenic protein that can be targeted by an antibody. So let's write all that out, man. Okay, so MHC-1. So nucleated cells. Display endogenous antigen. To, I'm gonna shorten it to CD8. plus cells. CD8 plus is just another way to label cytotoxic T-cells. It can be shortened to CD8 plus cells. It means the same thing, so I'm just doing that. And MHC2 So MHC2 is present in all professional antigen-presenting cells, such as macrophages, dendritic cells, some B cells, certain activated epithelial cells, and it displays an exogenous antigen, non-self proteins from outside the cell to helper T-cells. Helper T-cells, like cytotoxic T-cells, can be shortened to CD8 plus. Helper T-cells are shortened to CD4 plus. I will write all that out. So in displays exogenous antigen to CD4 plus cells. Again, CD4 plus meaning helper T cells. And we will get further into cytotoxic and helper T cells when we get into adaptive immunity. Anyways, moving on with the rest of these innate immunity cellular defenses. So, okay, dendritic cells, as I just mentioned, are antigen-presenting cells in the skin. So they have pattern recognition receptors that can recognize the category of the invader, which is important. It allows for production of appropriate cytokines. Remember, cytokines are responsible for inflammation. Appropriate cytokines to recruit the right type of immune cells. So let's write that. Oops. Nice to have a pen. Okay. Pattern recognition receptors. Receptors recognize category of invader. So, meaning like bacteria, fungus, parasite, or virus. Okay. And I suppose I'll just draw little arrows. These can go there or there. Alrighty, so natural killer cells, as I mentioned with our hematopoiesis diagram, they are non-specific lymphocytes. So they are still in the category of lymphocytes. They don't come from a myeloid stem cell, but they're still part of your non-specific immunity. So NK cells, they attack cells that are not presenting MHC molecules, including virally infected cells and cancer cells. So, it's important here. Yep. Okay. Now we'll get into the granulocytes. So, granulocytes, as I mentioned, include neutrophils, eosinophils, and basophils, and mast cells are like a sibling of basophils. So, neutrophils. Neutrophils ingest bacteria, so they're phagocytic. A particularly opsonized bacteria. Opsonization just meaning they've been marked with antibodies from B cells. And get into that later. So they can follow bacteria using chemotaxis, which just they navigate via chemical stimuli. So that's chemotaxis. Neutrophils are also the most populous of our white blood cells, and they last about 5 days. I think, yeah, they live about 5 days, and then dead neutrophils are what creates pus. Also, like if you have acne or regular pus in a wound, those are neutrophils. Those are dead neutrophils. So, anyways, let's see. Just bacteria. Follow using chemotaxis. I think that about covers it for neutrophils. Okay, eosinophils. They're used in allergic reactions and invasive parasitic infections. So they release histamine, which causes an inflammatory response. So, let's see. Allergic reactions. Okay. And another thing about histamine. So histamine, causes an inflammatory response, which results in vasodilation and increased leakiness, I'm going to say, of blood vessels. So additional immune cells, especially macrophages and neutrophils, can move into the tissue that is infected, which is useful against extracellular pathogens, mainly bacteria, fungi, and parasites. So I suppose I'll just write a little blurb on that. Use red. So histamine. Vasodilation. More immune cells into tissue. Oh, no. And, actually, yeah. I think I'm gonna redo this, though. That looks better. Okay. Alright, basophils are also used in allergic reactions, and they also release histamines, so and mast cells are like the sibling of basophils that are found in the skin. Tissues, mucosa, epithelium, things like that. I'll just say these are similar. Alright, moving on to the adaptive immune system. Whee! Okay, so adaptive, aka specific immunity. It takes time to activate. So instead of the innate immune system, which is always active, takes time to activate, but it can target a specific invader. So it can target a specific invader, and it can maintain immunologic memory. So this will all make sense in a minute. Takes time to activate. I actually have a kind of diagram going over how the adaptive immune system works as a whole, and it'll make sense why it takes time to activate. And we'll get into how it targets specific invaders and the immunologic memory part of it. So let's get going. So humoral immunity, if you remember, involves the B cells. So alright, so humoral immunity. it's centered on antibody production. So antibody production by plasma cells, if you remember those. If you remember, plasma cells are derivatives of B cells. Oh, which I have right here, which are activated B cells. Okay. So B cells mature in the bone marrow. B cells, bone marrow. Yes, that's how I remember that. So they mature in the bone marrow, but when they leave the bone marrow, they're still considered naive until they come across a specific antigen. And then they're like fully fledged B cells, I guess. So typically that means they're activated in the spleen or lymph nodes, part of your lymphatic system. And that'll be at the very end. I'll get into that a little bit. So activated in spleen and lymph nodes. Okay, and over here, antibodies, they act in the blood rather than cells, but they're also found in chyle, which is lymphatic fluid. I'll write that. Lymphatic fluid. And also antibodies can be found in the air. Okay. More on antibodies right now. So I have this labeled diagram of an antibody. So antibodies can also be called immunoglobulins, shortened to Ig, and I'll write that out in, let's do purple. So immunoglobulins. Immunoglobulins. I apologize. Also Ig. And there's several types of these that I'll mention in a second. So, I'm sorry, that was one of my fingers. Antibodies, their whole purpose is to target a particular antigen. All right, so they consist of 2 heavy chains, in these green colors here are heavy chains, and 2 light chains here in the blue. They have a constant region, which is, you can see, is this part of the Y, the constant region, which binds to natural killer cells, monocytes, macrophages, and eosinophils. All bind to the constant region of an antibody. And then there's the variable region at the very end here, and the tip of the variable region is the antigen binding site, where it'll actually attach to the antigen that it's seeking out. So variable region, one last thing on that, it's a very specific polypeptide sequence that will bind to only one specific antigen. So I said that the antigen binding site is, well, it'll bind to the antigen that it's seeking out. I said that, but really antibodies are kind of just like floating around until they come across the antigen that matches their antigen binding site. That's it. So, antibodies. When they're activated, the antigen binding region undergoes what's called hypermutation, which it improves the specificity of the antibody produced. So only B cells with high affinity survive, which is called the clonal selection. So I'll actually write that out. We'll do this in orange, actually. Yeah. Okay, so here's the bit on the different types of immunoglobulins. So cells can be given signals to switch isotypes. So they're called isotypes. I'll do it again in purple. Isotypes of antibody. So, and switching these isotypes is just stimulated by specific cytokines, and that's all you'll have to know about that. So there's, I'll do it in blue. For example, IgM, IgD, IgG, IgE, and IgA. Okay. And I'll write some little blurbs about what I said earlier when I was talking about all the different parts of the antibody. So with the heavy chain, the constant region, which is over here, constant region. Natural killer cells, monocytes, macrophages, eosinophils. Okay, great. And the variable region, gosh, I can't really write. Variable region. Only one. Only one. Okay. I think think that's all I'll need on that. So, okay. Now we'll get into, like, other functions of antibodies. So, like, when they're circulating in the blood, or in your lymph fluid, for that matter, they can, let's see, so I will label this in orange again. So they can opsonize, as we're getting into opsonization. They can opsonize pathogens, mark them for destruction. So they can cause they can neutralize pathogens if there's enough of them. So as you can see here, they're covering all the little binding sites on this pathogen, effectively neutralizing it. So and then they can clump several of them together. Several pathogens can all clump together with different antibodies, causing agglutination. So agglutination just means clumping. So they clump into insoluble complexes that are ingested by phagocytes eventually. So here I made a little drawing of, an opsonized pathogen that is then getting eaten. Yay! Okay. Cool. So now we're getting into B cells. So, memory B cells, they basically just lie in wait for a second exposure. I mean, I made this little diagram here. I suppose I'll use it. So, let's say you have exposure number 1 to a virus. We'll just say COVID. Let's be grim about it here. Let's say you have COVID, not once, but twice. Anyways, so first time you're exposed to COVID, you produce a certain amount of antibodies to help fight the infection, and then antibodies go down as the infection resolves. But then let's say you get COVID again, and because you already had the first infection, you've created memory B cells, which then will recognize the same pathogen. That's where memory comes into, immunologic memory comes into play in the adaptive immune system. And so memory B cells will recognize the pathogen, in this case COVID, and will mount an immune response not only faster, but stronger. So your serum antibody level goes up way faster and is way higher. So this is also the concept behind vaccination. So yay, vaccines. Cool. So yeah. So the point of a vaccine is to expose you to the antigen of whatever virus that you could come in contact with. Very, very good example is the annual flu. You could come in contact with whatever version of the annual flu is out, and if you had your flu vaccine, then you have already been exposed to a very similar influenza is really hard to get perfect, but very similar antigen, similar enough that you'll be able to mount a better immune response when you come into play with a real-life version. So that is active immunity, actually, which is, immunization. Vaccines are active immunity. I should have written that up there. Vaccines. Are active immunity. And, I don't know, while I'm talking about this, passive immunity is the transfer of antibodies to an individual from another individual. So, like, placenta and breast milk. Breast milk's a huge one, guys. My gosh. It's amazing the kind of stuff that's packed in there. Okay, so now we'll continue with cytotoxic immunity, also known as cell-mediated immunity, which is centered on the functions of T-cells. T-cells, which mature in the thymus. So we see a pattern here. T-cells, that's horrible. T-cells mature in the thymus. Okay. So, cytotoxic immunity center around the functions of T-cells. They directly kill virally infected cells. That's like their main function. Versus B cells, their function is to create antibodies, basically. Anyways, so T-cells, when they undergo maturation in the thymus, they do so via positive and negative selection. So positive selection selects for T-cells that can react to an antigen presented on MHC. So positive selection, say T-cells that Alright. And negative selection, it's just, so immature T-cells that are found to bind self-antigens are destroyed, apoptosis. So T-cells that bind self-antigens are destroyed. Via apoptosis. Cool. Alright, now, helper T-cells. Remember I talked about those as the CD4+, or also TH. So helper T-cells respond to the antigen on MHC2. So MHC2, and they coordinate the rest of the immune system. And they also secrete lymphokines. Lymphokines that activate various immune defenses. So lymphokines, they recruit plasma cells, TCs, which I will get into, and macrophages. Okay, I will explain this in a second. So, okay, Th1 cells, these guys, Th1 cells, they secrete interferon gamma, which activates macrophages. So which activates macrophages. And then Th2, they activate B cells, primarily in parasitic infections. Just a note, so they activate B cells. Okay, so in theory, what would happen if we didn't have helper T cells? Well, our immune system couldn't mount an adequate response to an infection, basically. So, immune system cannot mount response to an infection, which is exactly what happens with HIV. So, last little note, helper T cells, CD4+, are better at fighting extracellular infections. So, let's see. Oh, it's horrible. Better at fighting extracellular infections. Okay. Alright. Now, cytotoxic T-cells. TC, that's what that is. They're right here. So yeah, TC is cytotoxic T-cells, also known as CD8+, as was mentioned earlier. So, cytotoxic T-cells respond to the antigen on MHC1. And they kill virally infected cells. They do this by injecting toxic chemicals that promote apoptosis. Okay, And just as helper T-cells are better at fighting extracellular infections, cytotoxic T-cells are better at, ooh, I meant to say targeting here, not fighting, forgot to change that. Better at targeting. Good, intracellular infections. Okay. Fantastic. So, like I said, I mentioned a little bit earlier, autoimmune conditions are when a self-antigen is identified as foreign and the immune system attacks the body's own cells. Allergic reactions are non-threatening exposures that incite an inflammatory response. Actually, well, sometimes allergic reactions can be really bad, but I digress. Okay. So, moving on. So, suppressor or regulatory T-cells, they tone down the immune response after an infection. After infection, and promote self-tolerance. Self-tolerance. So they turn off self-reactive lymphocytes to prevent autoimmune diseases. Yeah, so if you have something wrong with your regulatory T cells, you're likely to develop an autoimmune disease. Okay. Last on this list is memory T cells, which serve a similar function to memory B cells. And again, I will get into that very soon. We're just going to briefly cover the lymphatic system. So, lymphatic system is a circulatory system consisting of one-way vessels with intermittent lymph nodes. So, circulatory system. Consisting of one-way vessels and lymph nodes. So the lymphatic system, it equalizes fluid distribution. When your lymphatic system is overwhelmed, this can result in lymphedema. I don't know if you've ever heard of that, but it's basically just like where your legs get super swollen, or even your arms, mostly your legs though, with lymph fluid that is overflowing from your lymphatic system. Okay. Also transports fats and fat soluble compounds in chylomicrons. microns, and also provides sites for mounting immune responses. Fantastic. So there is a pathway, I'm going to say, from how you get from the interstitial fluid, meaning the fluid between cells, all the way back into your bloodstream through the lymphatic system. So I'm just going to write out those steps really quickly. So it started the interstitial fluid, flows into the lymph capillaries, flowing into lymph vessels, going into your lymph duct, also known as your thoracic duct, so I'll put in parentheses here, thoracic, and finally ending up in your left subclavian vein. So which is just the vein underneath your left clavicle. That's all it is. All right. So here we go. This is what I was talking about. It's the last little bit here in this lesson. So here is a diagram that I drew out, a while ago, that shows the balance, I suppose, between the humoral immune response and the cytotoxic or cell-mediated immune response, or how they work together. So you have your first exposure to an antigen right here. So this antigen, free antigens directly activate your B cells, okay? And they also, the antigens displayed by infected cells activate your cytotoxic T-cells, but antigens can be engulfed by macrophages. Macrophages then become antigen-presenting cells, which stimulates helper T-cells. So then helper T-cells will also stimulate B cells, and they'll also stimulate cytotoxic T-cells. So there's it's a double whammy of how this antigen will get your adaptive immune system going. Then, so the helper T-cells, they also it's a positive feedback loop. And they also work with, they create memory helper T-cells. So memory helper T-cells come into play when you have a second exposure to an antigen. Before I get into that, so your B cells will give rise to two things: plasma cells, which secrete the bad boy antibodies that you'll need for several reasons, as previously explained. And they also secrete memory B cells, which also come into play in that second exposure to an antigen. Cytotoxic T-cells over here give rise to memory T-cells, and they also give rise to active cytotoxic T-cells. So, all right, let's go then to antigen your second exposure to this antigen. So it stimulates your memory helper T-cells, which then stimulates your memory T-cells, which stimulates production of active cytotoxic T-cells here, and then also stimulates memory B-cells, which stimulates more plasma cells to be made and more antibodies to be secreted, and it just these feedbacks is like that. So you can see that the I've done in green the first exposure, so all the arrows that are in green are involved in your first exposure to an antigen, and everything in purple is involved to second exposure to this antigen. So with humoral immune response, so their main job is to defend against extracellular pathogens by binding to antigens and making them easier targets for phagocytes. And then your cytotoxic immune response is responsible to defend against intracellular pathogens and cancer by binding to and lysing the infected cells or cancer cells. And so this diagram just gives you a little idea of how your adaptive immune system works altogether. Alright, and that is it, y'all. Thank you so much for watching, and I will see you next time. Bye!